Hello and welcome to another Piper Pod. As promised, part 2 of muscle physiology, we're going to look at the contraction itself. So last time we focused a lot on the anatomy, so the structure of it, all the structures that help us, and we talked about the stimulus and how different fibres are recruited. Okay, so now we're going to focus on what actually happens within the body when we go through a contraction. So it all starts with a motor neuron. Okay, motor neuron connected to the spinal nerves will receive a stimulus that we want to move. Maybe we want to pick up a water bottle to have a drink. Whatever it is, we get the signal that we want to move, and that will pass the signal down the motor neuron to the end to form what's known as a neuromuscular junction. Now, the neuromuscular junction, as we talked about, a motor unit, has multiple ends attracting with a whole bunch of different myofibrils. Okay, so that's what can happen with motor unit. It will essentially travel around the entire belly of the muscle, and it will stimulate a lot of different areas of the one. So it's not just sitting on top, it will actually surround and encapsulate. Okay, now we're not going to talk about how the messages pass through the neuron itself, okay, because that's the next lessons coming up on the peripheral and central nervous system. We're just going to focus solely down on the synaptic terminal and the synaptic cleft of the actual neuromuscular junction itself. Okay, so looking right down at that, as the impulse comes down, essentially gets to the action terminal, okay, the synaptic cleft, and the terminal is charged, if you will. Okay, so it receives positive energy, or it receives a membrane potential increases, which will open what's known as voltage-gated calcium channels. Okay, so they are calcium channels that are closed until it receives a certain amount of voltage. Okay, so as the voltage comes in, the calcium channels will open, causing calcium to come in to the actual synaptic terminal itself. From there, calcium, okay, whilst being a cation responsible for its own gradient of voltage charge, it's also an amazing electrolyte in causing second and third effects. Okay, so it actually will activate C proteins, activate enzyme responsibilities, and from there it will stimulate vesicles. The vesicles that contain acetylcholine, and it will force these vesicles to go to the actual edge of the synaptic cleft, okay, where we actually get to the junction, and it will force the release of acetylcholine into the synaptic cleft. Okay, and that's what passes it on. Okay, so acetylcholine is the neurotransmitter responsible in skeletal muscle contraction, and is passed on by enzyme or coenzyme responses stimulated by calcium itself. Once we get the vesicles to the edge of the actual membrane, it passes through acetylcholine. Now, acetylcholine will travel through the synaptic cleft itself, and it will bind to the post-receptive ports found on the sarcomere itself. Okay, it's what's known as the motor end plate, if you want to know. Okay, so as the acetylcholine passes through, it will bind to the receptor sites on the motor end plate itself. Now, the activation of these receptor sites will actually cause the membrane, the resting membrane, to charge, and that will open voltage-gated sodium channels, and the sodium will rush into the cell. Same time, potassium, in order to maintain equilibrium, will be drawn out of the cells, sometimes through the same channels, but other times through the additional channels that are open as a result of the higher voltage that you find at this membrane. Okay, so sodium comes in, potassium goes out, all because acetylcholine has touched this receptor site. From there, that membrane potential is passed on. So once the voltage of the membrane found on the motor end plate is large enough, it will spread out, opening more voltage-gated sodium channels, and then it will continue that action potential right along the sarcomere. Okay, remember as I described, burning a stick and it burns both ends, that action potential will pass on because of additional voltage gated sodium channels opening right along the membrane. So as it reaches that action potential, the gates will open, sodium will come rushing in, and then it'll open more gates beside it, all the way along, which will continue on that action potential. And we'll talk about what happens to that action potential during the next step. Okay, because what's happening at the same time as that is acetylcholine is being turned off. So it's getting picked up by an enzyme called acetylcholine esterinase, and that will actually pick it up and break it. And once it breaks it, it will return it back into the actual pre-synaptic axon in order to be instigated again. Now, a closer look at that process with acetylcholine. Okay, so just an overview and summary. We know the acetylcholine comes down, pushed into the synaptic cleft, activates the motor end plate, which opens sodium channels. Okay, from there, acetylcholine esterinase will pick it up and break it into acetyl, choline, and acetic acid. All these free compounds that cannot instigate another postsynaptic neural response will get transported back into the pre-synaptic terminal and be rebuilt and put back into vesicles waiting for the next stimulus. Now, if this acetylcholine esterinase didn't occur, then acetylcholine would be free floating around the area and it will be able to stimulate repeatedly this postsynaptic membrane. So what happens is, as acetylcholine comes in, it binds with it, opening up. Once the charge gets too great, acetylcholine is actually pushed off. Okay, it is pushed off the actual postsynaptic membrane, but then as it comes back down, acetylcholine can reactivate it and reactivate. Okay, and that happens when we're dealing with our cholinergic toxicities. Organophosphate poisons, funnel web spiders, those sort of things, it actually breaks the acetylcholine esterinase or binds with it so it can't pick up acetylcholine, which means acetylcholine is able to activate every time we get a decrease in that postsynaptic membrane voltage. And that will obviously give multiple stimulus. Hence why one of the symptoms is muscle twitching. Let's find out what happens with that membrane potential. Okay, where did that membrane potential take us? As promised, membrane potentials occurred along the sarcomere. It will travel along the sarcomere until it meets those areas that we spoke about in the first lesson called the T tubules. From the T tubules, it will travel down and it will activate the sarcoplasmic reticulum. Okay, so if we recall back to the microanatomy of the previous lesson, we have the T tubules surrounding all the actual fibres themselves, and then surrounding every myofibril is this specialized sarcoplasmic reticulum. As that voltage is passed on to the sarcoplasmic reticulum, that then will open all the voltage gated calcium channels, and calcium will be pushed out into the actual muscle itself, so into the myofibrils themselves, in order to be utilized for that sliding filament theory. Okay, looking at this sliding filament theory more closely, this picture here represents its resting. So there's no calcium involved, there's no action potential passed on, it is just as it is. So whilst it's in its resting stage, what happens is the myosin head have energy within them, ready to go, but the actin head is protected. Okay, so the receptor sites needed for the myosin head to connect are protected by those tropomyosin chains. Okay, so the tropomyosin chains will actually cover the receptor sites at rest. Okay, and the troponin will be sitting on top of them, just anchoring them in place. Okay, so that's what happens with a resting sarcomere. Once the process above that we just spoke about occurs and calcium is released from the sarcoplasmic reticulum into the sarcoplasm, then it is attracted and calcium will actually bind to the troponin itself. Now, as it binds to the troponin, it will actually change its voltage charge and cause the topomyosin chains to move. And as they move, it opens up the active site for the myosin head to bind to. Once stimulated by these receptor sites, the myosin heads can reach up and grab hold of the actual actin. Okay, and this is what's known as cross-bridge formation. Now this is what occurs during muscle tone. Okay, so we have a tightening, we have a fusing of the cross bridges. This is also all that happens during your isometric contractions. These large contractions, you just get a full cross-bridge formation. So you get that strength, you get the holding power, the inability to stretch the muscle further, but there's no shrinking at this stage. Okay, so that's cross-bridge formation. From there, we go into the pivoting of the myosin head. So as a result of this, the myosin head loses its ATP and phosphate, and it contracts and pushes the actin towards the center of that M line that we spoke about. Okay, so it's pushed it in. Now this myosin head will actually stay connected to the actin until it's triggered to be released by another ATP. Okay, so this process here is actually what causes your rigor mortis in people. Okay, calcium will be flowing through the body, which will activate it. Myosin will be able to grab hold of the actin, pivot the myosin head in, causing a tightening of the muscles, but then it won't be able to be released. Okay, because there's no free-flowing ATP when you're dead, therefore you end up getting rigor mortis. So we've lost an ADP and phosphate. Calcium's still bound to the troponin, and we're able to push the actin closer to the M line. From there, ATP comes along, and once ATP comes along, the myosin is able to go through what's called cross-bridge detachment, and then lower back down, ready to pick up the next grab. It's been released from the actin, and then it's ready to be activated again. Once we go through reactivation, the myosin head hydrolyzes the actual ATP, and it gets ready to be reactivated as it goes through, as it finds another actin receptor site that's open, and the same process goes through each time. Okay, so that's how the whole process works. Now, looking at it very simply like this, it makes it easy to think that this whole contraction is based on one movement of the myosin head. So the myosin will lift up, hold on, move it, and that's what the contraction is. Okay, but actually it's a series of these processes going through, where the actin will be in place, the myosin head will be able to bind with it, pull it, get released by ATP, bind with it again, pull it again, and it actually keeps doing it, keeps pulling the actin in several hundred times. It actually will happen before a contraction can be fused. Now the reason it's called the sliding filament as opposed to the ratchet filament, or every time you reach up and pull it and the myosin releases, we don't lose the actin all the way back because of the tightens that we talked about, is because we have hundreds of myosin heads, and each of them are not released and activated at the same time. So because we have this partnering coupling effect, we're able to pull the actin along, hand over hand if you will, until we reach optimal contraction required, and then once we all release, we'll talk about what happens when relaxation occurs. Okay, because right now relaxation will never occur as long as calcium is still present. Myosin will keep binding to the actual actin head, even if it doesn't pull it anywhere. It will just keep pulling it into the center and then releasing each time, burning up energy as we go along. Okay, that's why when we hold a maximum contraction, we still get fatigued. Okay, because whilst we're holding that contraction, calcium is still present, which means ATP is still being utilized to detach the myosin, reattach the myosin, detach the myosin, reattach the myosin, but the actin is not going anywhere. Okay, that's why you can't hold a 30 kilo plate in bicep contraction indefinitely. It's because of the fatigue and most of the time the build-up of lactic acid required to keep up with those energy reserves. So what happens when the muscle relaxes? Obviously left-hand side is a overview of what we just spoke about. Nice and simple. Presence of calcium, myosin, pull the actin in together. Contraction starts. When relaxation is needed, acetylcholine is broken down with acetylcholine estranase, so we're no longer getting that stimulus. There's no longer a stimulus coming down, therefore there's nothing activating the T tubules, the sarcoplasmic reticulum gates are not being open, so it goes through a process, active transport of reuptake of calcium. Now a little bit of trivia for you is actually the reuptake of calcium into the sarcoplasmic reticulum that generates the most heat in your body. That is why you get really hot when you exercise, is because of the heat generated by the uptake of the sarcoplasmic reticulum. Okay, just as trivia. From there, no calcium, the topomyosin that we spoke about goes back to its resting position, and no actin head receptor sites are available for myosin. So no myosin attachment. From there, the titans that we spoke about assist in the relaxation of the muscle back to its resting position. And that's what it goes through. That is the steps in a nutshell for relaxation. Now, if we think about what here needed ATP, okay, just to break it down, we obviously need ATP for the myosin heads to connect and detach. Okay, ATP is needed mostly for the detachment, same one's needed for the connection as well. Okay, as it's broken down into ADP and phosphate needs to attach. So that whole process there, every time it goes through and drops, it's needed. Every single time. Remember I said before you get a full contraction, that occurs hundreds of times for every single muscle, every single sarcomere. It happens hundreds of times. Every muscle fiber has numerous sarcomeres, and every muscle has numerous muscle fibers. Okay, so that's needed for that. Each myosin head needs an ATP each time it moves. On top of that, we need active transport to help in this process. So the reuptake of calcium is done by active transport. So that actually takes energy to do. ATP is needed for that to occur. On top of that, we also have this whole amount of sodium and potassium on the wrong side of the membrane. So the resting membrane potential has been altered. So the body has to correct that. Now, yes, we do have a couple of open potassium changes that can push it out, but most of the time, it's the active transport of the sodium-potassium pump that will cause the swapping of one potassium to sodium each time it goes through. And that takes ATP as well. So this whole process will take thousands and thousands, if not millions, of ATP to go through for a muscle to fully contract and relax each time. Okay, so now when we think about why we can only last 15 seconds for creatine phosphate, why we can only last about 40 seconds for anaerobic glycolysis, because we're not talking about it on a simple level where 2 ATP is, well, that's a good amount. We're talking about it happening thousands of times and utilizing millions of ATP for a simple contraction. That is going to build up a lot of lactic acid in the body, not just one molecule, but thousands of molecules of lactic acid, which is why eventually we'll need to stop. Okay, so that is physiology of how we contract. The whole process that it goes through from the initial stimulus right through to the sliding filament theory. I hope you got something out of it and have a little bit of better appreciation of what happens every time we move. But next lesson for the anatomy physiology, as promised, we'll be focusing on the peripheral nervous system itself. So until next time, take care.